Hi everyone. In this video we're going to be talking about the loop of Henle. So think of it as a bit of a continuation from the proximal convoluted tubule. So the fluid has moved into the loop of Henle, and first we'll just sort of review the basic structures of the loop of Henle. So first we have the descending limb, and then we have the hairpin turn at the very bottom of the loop of Henle, and then we have the ascending thin limb and the ascending thick limb. Now, the descending limb, hairpin turn, and ascending thin limb are all made of simple squamous epithelial cells, which means one single layer of the flat squamous type cells, and that basically is, although this is a two-dimensional drawing, around the entire renal tubule. And the ascending thick limb is made of simple cuboidal epithelial cells. So these cells are a little bit bigger, and as we get to each segment, we're going to draw in the different types of transporters that are present in those locations. So we'll also draw in really quickly the vasa recta. And remember, the vasa recta is the primary capillary type vasculature that is found around the loop of Henle, particularly in the juxtamedullary nephrons. So not every nephron is going to have a vasa recta. But what we're drawing here basically is the loop of Henle that descends way into the medulla of the kidney, and so the vasa recta is just going to follow sort of around that. Now, in the descending limb, it's permeable mostly to water. So we're going to draw in a bunch of aquaporins, and the aquaporins are going to allow for water to travel from the tubular fluid through the epithelial cells. So there's going to be aquaporins at both the apical and basolateral surfaces, and then the water will move into the ECF, which is alternatively called peritubular fluid, but I'm going to just label it as ECF. So these aquaporins allow for passive transport of water, and the passive transport of water is so certainly it's going to move into the vasa recta, but also it's going to accumulate in the extracellular space, and that's going to be important later on. So we can think of a bunch of water being present and then sort of diffusing outward in all directions so that it becomes moderately evenly distributed in the extracellular space. And we'll draw that in just a few moments. A couple of other things that can be reabsorbed in this location are sodium and urea. So both of these it's a very small amount of reabsorption relative to the amount of water. And really that's just because the water moves so much faster. So we could think of this as primarily water, and then just a little bit of sodium and urea as well. So let's just talk a little bit about the osmolarity of the fluid here and what's happening. So we're coming from the proximal convoluted tubule with an osmolarity of 300, and we know that in the proximal convoluted tubule we had reabsorption of both water and solutes, about 67% of the total, but in the ratio that sort of maintains the osmotic concentration. And so as we move into the loop of Henle, we're going to start to change that a little bit. And so moving from the proximal convoluted tubule down the descending limb of the loop of Henle, we're reabsorbing water primarily in very, very small amounts of solute, meaning that in relative proportion, at the hairpin turn, there's going to be significantly more solute than there will be water. And just for the sake of comparison, the osmolarity can be as high as about 1,200 to 1,400 milliosmoles per liter in the very base of the loop of Henle. So what this means is, as we approach the ascending thin limb of the loop of Henle, we're going to have a now higher solute concentration. So let's just draw in. We mentioned earlier that the water that was reabsorbed from the descending limb is going to diffuse and actually sort of spread out throughout the entire extracellular space within the medulla around the loop of Henle. And so in comparison, there's going to be a high water concentration in the ECF because we have a lot of water that was reabsorbed that's moving, and because we have less water in the loop of Henle, we're going to have a high solute concentration in the renal tubule of the ascending thin limb. So in the ascending thin limb, there are transporters, most likely co-transport, but a lot of times this isn't really specified. It's more just definitely passive diffusion of both sodium and chloride. So it's going to be through both the apical and the basolateral surfaces. And so now as we reabsorb the salt, we're going to see the salt sort of do the same thing that the water did. It's going to naturally want to spread out around the entire ECF, which means that, among other things, it's going to end up moving towards the descending limb. So one critical point here is that while the salt is moving, there is no water movement. And this is going to be true for both the ascending thin limb and the ascending thick limb. There are no aquaporin channels in either location. And this is necessary because if there were aquaporin channels, then we wouldn't necessarily be able to maintain our salt gradient and have the salt move. So if there were aquaporin channels, what would happen most likely is the water would actually move back into the renal tubule, and because now the solute concentration is higher. And so I think the thing to really kind of keep in mind is that when we talk about movement of salt or any type of solute down its respective gradient, it's going to move from high to low. But if we mix that with water wants to move from high water to low water. The water just is faster than the solute. And that's the same thing as what happened in the descending limb. The water moves fast and the solute moves slow in comparison. So if you have two separate spaces that are permeable to both salt and water, you're going to see the water move and sort of equilibrate the both environments before you're going to see the salt do that. And so in order for this to work, in order for us to only reabsorb salt in the ascending limb, it can't be permeable to water. So you're going to see no aquaporin channels. So as we move from the ascending thin limb to the ascending thick limb, we're actually going to see a different sort of mechanism for salt transport, but the same situation occurs. We're going to move the salt, and of course, we can't move the water. All right, in the ascending thick limb, we have a particular transporter, which is named the sodium potassium 2 chloride transporter. It's a co-transport mechanism that moves 2 chloride ions and 1 potassium ion and 1 sodium ion from the renal tubule into the renal epithelial cell. So it's only located in the apical surface of the cuboidal cells of the ascending thick limb, and it's passive. It doesn't require any energy to function, but it's going to move all those things into the renal epithelial cell. And then from there, and just because there's not really enough room in this drawing, so we'll just sort of draw them separately. From there, we're going to move there's going to be co-transport for the chloride and the potassium ions. So 1 chloride and 1 potassium will move together from the epithelial cell into the ECF. And then similarly, there will be a sodium-potassium pump, and the sodium-potassium pump will exchange the sodium ion that is in the epithelial cell that needs to move into the ECF for 1 potassium. So what this means in summary is that we are going to use the sodium-potassium 2 chloride transporter along with these other transporters in the basolateral surface to help us reabsorb sodium and chloride. What we're not doing is reabsorbing potassium here, and not to say that we can't control the secretion or reabsorption of potassium, because we do, but this particular mechanism, when you combine all three carrier transport proteins, is actually sort of just exchanging the potassium so that we can move the sodium and the chloride where we want to. So we'll address the potassium separately. And finally, just in the same way that we had the sodium that was reabsorbed sort of diffuse outward and travel its way towards the descending limb, the sodium that's reabsorbed here in the ascending thick limb is also going to follow that same scheme. I think the thing to keep in mind, major difference between ascending thin and ascending thick is that ascending thin is passive reabsorption of sodium and chloride, and ascending thick limb is active reabsorption of sodium and chloride because we need that sodium potassium pump in order for it to work. But generally speaking, otherwise we can just sort of consider them as overwhelmingly their job is to reabsorb the salt. And that will help us sort of see this in the big picture. So now that we've made our way through the descending and ascending limbs, let's just talk about the sort of overall event that is happening here. And this is probably the most critical point of what the loop of Henle is capable of. It's called countercurrent multiplication. And it's essentially kind of like a positive feedback cycle that's occurring here. So we have water reabsorption in the descending limb, which in turn is going to cause an increase in solute concentration in the ascending limb and an increase in the solute gradient to the ECF in the ascending limb as well. So high solute in the renal tubule and lower solute relative to the inside in the ECF. So the result of these two things is that we're going to get an increase in sodium and chloride reabsorption in the ascending limb, passive in the thin limb and active because we need to use the sodium potassium pump in the thick limb. And so as we add additional sodium and chloride into the ECF, we're now going to see that diffuse towards the descending limb, which is going to further increase the gradient for water to move from the renal tubule into the ECF. So that process, you could just sort of say that over and over. And ultimately what we have is the ability to control water and salt reabsorption independently. So I just want to make a couple of final points as we wrap up. So as the tubular fluid is moving out of the ascending thick limb of the loop of Henle, its next destination is going to be the distal convoluted tubule. And unlike what has happened up to this point, we had 300 milliosmoles entering from the PCT, and we essentially concentrated the urine as we moved down into the base of the loop of Henle around the hairpin turn. But as we're leaving, even though we're reabsorbing the salt in order to maintain the countercurrent multiplication process, we are going to be leaving the ascending thick limb with a relatively lower solute concentration. And this is dependent upon multiple factors, but could be as low as about 100 milliosmoles per liter as it enters into the distal convoluted tubule. And that's something that we'll follow up in the DCT as we talk about that in our next video. The last thing I want to mention before we move on is to discuss the vasa recta briefly. So we didn't talk too much about it, but one thing that's important about the vasa recta is that unlike some of the other vasculature, the vasa recta sort of mirrors the osmolarity of the ECF. And this is a critical point because we want to ensure that the ECF sort of stays very, very concentrated in the base of the medulla, so towards the bottom of those renal pyramids, just before the urine sort of leaves and is going to enter into the renal pelvis. We want to make sure that it stays concentrated in order to maintain countercurrent multiplication. If we had fresh low osmolarity blood, or sort of even baseline osmolarity, 300 milliosmoles per liter blood traveling through the vasa recta, then essentially it would wash away the gradient that's been generated from the top of the loop of Henle to the bottom of the loop of Henle, and the matching gradient in the medulla, in the ECF. So the vasa recta actually is going to mirror the osmolarity in order to maintain countercurrent multiplication. So it's just one point that is kind of minor, but really speaks to the significance of the vasa recta as opposed to just having peritubular capillary taking up all of this space. So that concludes our discussion on the loop of Henle. And once again, moving forward, we'll be talking about the DCT and the collecting duct. So I hope you enjoyed it. Thanks for watching.